Our next researcher will be Gillian Cameron, and she will be talking about evolve, rinse, repeat, the evolution of copper resistance in Legionella pneumophila.
Legionella pneumophila is almost 2 billion years old, but as a pathogen of amoebas, it hasn't really been a problem for humans, at least not until the past 50 years. Legionella causes Legionnaire's disease, a severe form of pneumonia with a 10% mortality rate. It spreads to humans when water droplets containing the bacteria are aerosolized and then inhaled. Now, nature doesn't aerosolize water very often, but we do. Our engineered water systems, like air conditioning and even showers, create the perfect growing environment for amoeba and the Legionella that infects them. Copper-silver ionization, the process of using metal ions to kill bacteria, is one method for Legionella control. Copper is great at killing Legionella. Well, it's great at killing most Legionella. We're starting to see the development of Legionella strains that are resistant to this treatment. But why? Well, copper pipes and engineered water systems act as a selective pressure on the bacteria. Over time, these bacteria evolve to become resistant to copper and are able to survive treatment with copper-silver ionization. When some of the genomes of these resistant isolates were analyzed, it was found that they differed from their copper-sensitive counterparts in the same water system by only 29 individual nucleotides. So, how are these strains becoming resistant to copper? That's where I come in. Hi, my name is Gillian. I'm a PhD student in the Faucher lab, and I hypothesize that copper resistance in Legionella emerges as the result of the accumulation of individual point mutations in response to persistent exposure to copper. To find if this is the case, I am generating copper-resistant lineages of Legionella in the lab in a process known as experimental evolution. I grow up cultures of Legionella, I wash and resuspend the bacteria in water, I expose the bacteria to copper, and I grow the survivors. I will repeat this process dozens of times, exposing successive generations of bacteria to increasing concentrations of copper ions, until eventually I will have copper-resistant lineages of Legionella. I will then sequence and analyze the genomes to identify any mutations that may confer copper resistance. To confirm the effect of these mutations, I will induce each point mutation into the ancestor of the evolved lineages. I will then expose the resulting mutants to copper to examine the effect the point mutation has on copper resistance. Infections with Legionella are increasing worldwide, meaning it's more important than ever to find ways to limit the spread of Legionnaire's disease. This starts in the engineered water systems. By understanding how copper resistance emerges in Legionella, we can more effectively deploy metal-based methods of bacterial control, preventing people from getting sick and avoiding the development of resistance in the first place. Thank you.